Human interactions with bacteria. Human-bacteria interactions. Humans interact with bacteria in a variety of ways. Symbiosis, pathogenesis, and fermentation are types of interactions that will be explained on the following slides. Symbiosis. Bacteria cover every surface of our environments, our skin, and our digestive tract. We have a mutualistic relationship with most species. We need them to maintain healthy skin, digestion, or hormonal regulation, and the bacteria also benefit by living with us and other microbial species. This community of bacteria is part of the human microbiome, the complex community of microorganisms with which we coexist. A balanced microbiome is a crucial factor for good health. Pathogenesis. Bacteria can make us sick via a variety of mechanisms. If we become infected with a bacteria that is usually found in other species, it may make us sick. These are zoonotic infections. Some zoonotic bacteria make us sick because they release toxins, chemicals that cause damage in human bodies. Examples of pathogenic bacteria that release toxins are Bacillus anthracis, which causes anthrax, Clostridium botulinum, which causes botulism, and Yersinia pestis, which is responsible for the plague. Zoonotic diseases spread between animals and people. Different types of bacteria have different modes of transmission. Pathogenic bacteria can move from animals to people by direct contact, by touching the same objects, by being bitten by an insect carrying the bacteria, or by consuming contaminated food or water. Occasionally, neutral bacteria can make us sick if our microbial community becomes unbalanced. For example, if we receive a high dose of pathogenic bacteria that our bodies usually control, or if a resident bacteria begins to replicate in a new place such as the bloodstream or urinary tract, these bacteria can make us sick. An example is E. coli, a normal inhabitant of our digestive tract. Some strains of E. coli, however, can make us sick if our food is heavily contaminated with it. Other strains of E. coli can multiply in our urinary tract or bloodstream and can be very dangerous in those organ systems. Fermentation. Almost every human culture uses strains of anaerobic bacteria as tools to ferment foods such as soy, maize, and milk. What is fermentation? As a reminder from the presentation on energy, fermentation is a type of cellular respiration that takes place in the absence of oxygen. Anaerobic means without oxygen. The types of fermentation used to preserve food depend on local customs and ecology. Lactic acid fermentation starts with glucose entering the glycolysis pathway. Pyruvate does not enter the mitochondrion, however. Rather, the fermentation of pyruvate produces 4 ATP, 2 NAD+, and 2 lactates per molecule of glucose. This process is used to preserve many foods, including milk, and often uses bacteria of the genus Lactobacillus or Lactococcus. When these bacteria partially digest milk or cream, the products are easier for many humans to digest and will last longer before spoiling. The table on the following slide lists many strains of Lactobacillus and other microbes commonly used to ferment traditional foods in Africa. Fermented foods have been a part of the human diet since the dawn of human civilization, and fermentation has been used as a means of improving shelf life, safety, digestibility, and nutritional value for more than 6,000 years. Food and Nutrition Research, 2016. For citation and further information, including the table, visit the website URL shown here. This table summarizes fermented products that are produced in various regions of Africa with various substrates that are involved in making those products and the microorganisms that are used to make those fermented products from those raw substrates. Alcoholic fermentation starts with glucose entering the glycolysis pathway. Pyruvate does not enter the mitochondrion, however. Fermentation of pyruvate produces 4 ATP, 2 NAD+, 2 ethanol, and 2 CO2 per molecule of glucose. This process is used to produce bread, wine, and whiskey, and often uses unicellular fungi or yeast, such as S. cerevisiae. Fungi will be discussed in the next section of this presentation. Technology. In addition to using bacteria to preserve and enhance some foods, humans use bacteria as a tool in biotechnology. Genetic engineers modify bacteria and culture them to produce large quantities of therapeutic proteins. We cannot produce insulin for diabetics without the use of bacteria in the laboratory. Bacterial cultures can be organic factories for medicines. Biotechnology will be further explored in the next presentation. Observing bacteria. Individual bacteria cells can be seen with a classroom light microscope that has at least 400 times, preferably 100 times, power. The next 3 slides show microscopic images of bacteria. Observing cultured colonies of bacteria will be discussed in the next presentation. This image shows cocci at 1,000 times. This image shows bacilli at 1,000 times. Yogurt contains lactobacillus. It may be possible to see it under a classroom microscope. The image below shows spirilla at 1,000 times.